Hello, my name is Anata Bezman, and I'm a sophomore in high school from the Bay Area. My research was called Revolutionizing Gene Editing with siRNA, saRNA, miRNA, and the CRISPR-Cas systems. I chose to research this topic because I'm deeply interested in the field of gene therapy, which can occur at the intersection between biology, engineering, and medicine. So, what is gene therapy? Gene therapy is a medical technique that can treat or prevent diseases by directly modifying gene expression. Gene expression can be modified through upregulation or activation, silencing, and correction. Upregulation occurs when the transcription of an essential gene is activated to increase the expression of that gene. On the other hand, silencing is the degradation of mRNA, thus preventing the gene's expression. And for correction, a mutated gene is altered to restore its normal function. Out of the many techniques in the world of gene therapy, I chose siRNA, small interfering RNAs, saRNA, small activating RNAs, miRNA, or microRNAs, and the CRISPR-Cas system. Before we dive into the actual mechanisms utilized in gene therapy, it's essential to understand the central dogma of biology, which explains the flow of genetic information. This idea is depicted through this flowchart in the slide. First, DNA contains the genetic information that is then transcribed into an RNA or mRNA molecule. This RNA sequence is then read by organelles called ribosomes, which use that information to assemble a functional protein. So, because mRNA is the intermediate between DNA and proteins, targeting RNA to either block the production of damaging proteins or enhance the production of beneficial ones can then alter the proteins being made. Now, let's get into the actual mechanisms of each of these techniques. So this first mechanism is the mechanism of small interfering RNAs. So the first step is the enzyme dicer cleaves the double-stranded DNA, generating these small fragments, which are 21 to 25 nucleotide long strands. The RISC, or the RNA-induced silencing complex, then takes one strand, or the guide strand, as shown here, and identifies and binds to complementary RNA sequences inside the cell. By doing so, it cleaves and degrades the target mRNA, silencing its expression. The second mechanism is miRNA. So this mechanism is a little more complicated than the mechanism for siRNA, as there are two routes depending on the degree of complementarity between the mRNA and the mRNA that it binds to. So for high complementarity, the 5' cap is removed, the poly A tail is shortened, all destabilizing the mRNA, and then further breaking down of the mRNA. For partial, there's four possible methods. It's not known exactly which one it is, but there are four possible methods of inhibition. So the first method is the competition for the 5' binding site between this miRSC complex and another protein complex. The second is the disruption of mRNA circularization and therefore stability by this CCR4 knot complex. The third is the prevention of the 60S and 40S ribosomal subunits from joining each other by the miRSC located in between them. And fourth is the accumulation of mRNA in P bodies. So in both of these cases, because mRNA is being broken down or translation is repressed in the case of partial complementarity of miRNA, proteins cannot be made, effectively preventing the gene's expression. Now for activation, this is a diagram of the saRNA mechanism. So the first step is the antisense strand is loaded by the Argonaut 2 or AGO2 protein, therefore emitting the sense strand. Subsequently, AGO2 loads with the antisense strand and then recruits two other proteins, RHA or RNA helicase A and CTR9, that interact with RNA polymerase II to form this complex called the RITA complex or the RNA-induced transcriptional activation complex. By this complex binding onto the mRNA, it drives transcription and elongation, further promoting the creation of proteins and increasing the gene's expression. Lastly, this is a diagram of the mechanism followed by CRISPR-Cas, which are involved in correction. So there's three stages of this mechanism: acquisition, which is shown here, where the cell recognizes this invading DNA and extracts a segment called a protospacer. This protospacer is integrated into the CRISPR array as a new spacer, as shown here, and is guided by the presence of a PAM, or a protospacer adjacent motif. So now here is the new CRISPR array, and in the next stage, biogenesis, this CRISPR array is transcribed into a long precursor CRISPR RNA, and this precursor CRISPR RNA is then further processed into small guide cRNAs, each containing one spacer, as shown here. And the last stage, interference, it varies based on the class of the CRISPR-Cas system, as class 1 uses this multi-protein complex called cascade to interact with the CRISPR RNA and therefore interfere with the nucleic acids, while class 2 just uses one effector protein to interact with the CRISPR RNA and then invade the nucleic acids. So now that we've looked at the different mechanisms of gene therapy, it's important to acknowledge and understand some of the key challenges for each of these techniques. So two of the biggest hurdles that need to be addressed are delivery and off-target effects. So let's start with delivery. RNA-based therapies generally face a lot of stability issues in the body, as they are more prone to degradation by enzymes. So in this diagram, there's an RNA, siRNA, mRNA, siRNA, doesn't really matter as all three are RNA-based, being degraded by a nuclease. So for a therapy to be effective, it needs to reach its target cell. And because the RNA is being degraded, it is then unable to reach its target cell and be an effective form of treatment, as shown here. The second challenge is off-target effects. So with these therapies, as they are right now, there are also high chances for off-target effects to occur, as these techniques often bind to incorrect regions with high complementarity to the target. So as shown in this diagram, here is the intended site, but the RNA is binding to an off-target site that's not the intended site, that may just have high complementarity as well. When this happens, there could be a lot of issues, such as unwanted gene silencing, unwanted gene activation, or just mutations, all effectively leading to harmful effects. So, what about applications? One example is through siRNAs, which are currently being explored as a therapeutic approach for ischemic stroke. So, the central goal of this technique is restoring lost neurological function. Generally, stroke leads to all kinds of problems, such as inflammation, apoptosis, which is cell death, and oxidative stress, all contributing to brain cell damage. And what siRNAs can do is they can actually silence key protein pathways activated post-stroke. And there's two different ways that they could do this, which are neuroprotection and involvement in neurodegenerative mediation. So starting off with neuroprotection, let's say this is our ischemic area. So specific siRNAs are actually able to target proteins that contribute to cell death and injury after injuries in this area. So for example, ASK1 siRNA suppresses upregulation of ASK1, which is a protein that promotes oxidative stress-induced apoptosis. So therefore, the reduction in this protein can prevent apoptotic cell death after injury. Another example is CHOP siRNA, which downregulates CHOP, which is a stress-induced protein that activates caspase-3, a major driver of apoptosis. And caspase-3 siRNA also silences expression of caspase-3, which, as I just said, is a major driver of apoptosis. So, researchers have actually observed improved motor function and a significant reduction in neuronal apoptosis when using these three methods. Then for neurodegenerative mediators, so generally neurodegenerative mediators are proteins that contribute to secondary damage and cell death after stroke. So what Beclin-1 siRNA can do is it can inhibit Beclin-1, which is a protein involved in the process of autophagy, which is the process of the body breaking down and recycling damaged components. By blocking autophagy, it enhances neuroprotection, thus preventing neuronal loss. For the sake of time, this will be the only therapeutic application I will cover, but feel free to ask about any other applications later if there are any questions. So, key takeaways. Gene therapy itself is a promising avenue for tackling a wide range of diseases by targeting the genetic root causes. Unlike traditional treatments, gene therapy holds the potential to offer long-term solutions and even cures by addressing the disease at the genetic level. Despite these challenges, ongoing advancements and research are critical. As we refine these technologies, gene therapy can essentially revolutionize the way we treat and even cure many previously untreatable conditions. Thank you so much for listening. If you have any questions, please don't be afraid to ask.
Great job, Anata. Thank you so much for your presentation. I really loved all of the illustrations too, and the way that
Mhm.
you
Thank you.
communicated those things. That was great. Cool. So yes, as you said, feel free to put anything into the chat. I have a few questions of my own. So first question from me is, of the different mechanisms that you've described, are there some that are more popular than others that people are pursuing as kind of therapeutic approaches to things, or are they kind of all just being explored right now pretty equally?
I mean, as I said, each of them do different things. So, so they can all be used to treat all sorts of diseases. So, for example, cancer is currently being looked at through siRNA and miRNA, and saRNA, depending on what they kind of want to do with it. Um, CRISPR is really popular right now with just genetic disorders that occur due to issues with one mutation, such as sickle cell disease. Where sickle cell disease, there's just one mutation, where with CRISPR, you can fix it and therefore fix sickle cell disease. So that one's really popular for genetic disorders. The other ones are more popular for disorders such as cancers, where you want to increase um, the expression of tumor suppressors or decrease the expression of genes promoting cancer. So all of them are being looked at for different applications a lot right now, especially since these are really new advancements.
Yeah, awesome. That makes a lot of sense. Great. Well, thank you so much. That was super informative. And I really appreciate it. All the work you put into it. It was really impressive.
Thank
So,
you.
tell me a little bit about your interest, and is this something you want to kind of continue to study more, or what's kind of next for you with your exploration of this?
So next, I kind of want to take this knowledge, this like foundational knowledge that I learned, like the knowledge of how they work, what some challenges are, and maybe see if I could use it more hands-on. So maybe go into a lab and use CRISPR to kind of cause some kind of change in some kind of genetic sequence and see what that does. So I guess just using this foundational knowledge kind of more hands-on to see if I could, you know, make maybe contribute to some kind of new discovery in the world of gene therapy, or just learn more hands-on about how gene therapy works, how it can be utilized, stuff like that.
Yeah, makes a ton of sense. And you have a great foundation from what you presented. It's really impressive. All you know already, and wherever you go next, if you do get it in a lab to do this, they're going to be really impressed by what you know already. So great job. Thank you so
Thank
much.
you.